Introduction. Welcome to today's lecture on the fascinating topic of spliceosome assembly and alternative splicing. As medical students, it is crucial to understand the intricate mechanisms behind gene expression regulation, particularly the process of RNA splicing. This process plays a significant role in generating functional mRNA molecules from pre-mRNA, thus leading to protein diversity. Throughout this lecture, we will delve into various aspects of splicing, including its association with disease and the therapeutic strategies available for correcting splicing-related disorders. So let's begin our journey into the world of molecular biology at the spliceosome assembly. The spliceosome is a complex molecular machine that removes introns and joins exons to form mature mRNA. Its assembly involves specific recognition of splice sites and stepwise recruitment of snRNPS and RNA-binding proteins. This precise process ensures accurate mRNA splicing. Alternative splicing is a process that allows a single pre-mRNA molecule to create multiple mRNA versions with different functions. This increases protein diversity, which is important for regulating gene expression and can affect cellular processes. Splicing factors interact with regulatory elements to control the inclusion or exclusion of specific exons, ultimately determining the final mRNA composition. Studying alternative splicing is crucial for understanding gene regulation and its impact on diseases. Intronic splicing enhancers, ISs, and silencers, ISSs, are regulatory elements within introns that interact with RNA-binding proteins. These proteins, such as SR proteins and hnRNPS, can either enhance splicing or inhibit it by influencing splice site recognition. Dysregulation or mutations in these elements can lead to abnormal splicing and disease. Splicing plays a crucial role in gene regulation, and its disruption leads to various human diseases. Mutations in splicing factors or splice sites can cause diseases like spinal muscular atrophy and cystic fibrosis. Understanding these connections opens doors for therapeutic interventions. In summary, splicing regulation is a complex and dynamic process involving various splicing factors and their interactions with RNA and proteins. Understanding the dynamics of these factors is crucial for developing targeted therapies to treat splicing-related diseases. Small nuclear ribonucleoproteins, snRNPS, composed of small nuclear RNAs, snRNAs, and proteins, are essential for spliceosome assembly and catalysis. Different snRNPS play specific roles and contribute to spliceosome stability, activity, and base pairing interactions. Studying snRNPS is vital for understanding splicing and addressing splicing-related diseases. RNA-binding proteins, like SR proteins and hnRNPS, regulate splicing by binding to specific sequences. They either enhance splice site recognition or inhibit it, influencing spliceosome assembly. Dysregulation of these proteins can contribute to human diseases by causing abnormal splicing patterns. The spliceosome is a complex that catalyzes the precise removal of introns and joining of exons in mRNA. It undergoes conformational changes to facilitate two reactions, resulting in excision of introns and release of mature mRNA. Dysregulation or mutations in spliceosome components can cause diseases. The dynamic nature of alternative splicing has played a significant role in genome evolution. The emergence of alternative splicing has allowed organisms to expand their proteome complexity without requiring an increased number of genes. Alternative splicing has enabled the generation of diverse mRNA isoforms, contributing to the adaptation and phenotypic diversity observed in different species. Furthermore, alternative splicing events can create new protein functions, regulatory mechanisms, and cellular processes. These innovative splicing events have contributed to the complexity of multicellular organisms and the evolutionary advantages they possess. Exploring the interplay between splicing regulation and genome evolution provides valuable insights into the remarkable diversity and complexity of life. Emerging evidence suggests that alterations in splicing regulation and splicing factors contribute to cancer development and progression. Dysregulation of splicing can impact crucial cellular processes, including cell cycle regulation, cell proliferation, apoptosis, and invasion. Mutations or aberrant expression of splicing factors, such as serine arginine-rich, SR, proteins or heterogeneous nuclear ribonucleoproteins, hnRNPS, can disrupt normal splicing patterns, consequently affecting gene expression and protein functions. These changes can lead to uncontrolled cell growth, reduced cell death, and enhanced metastatic potential. Understanding the role of splicing factors in cancer opens avenues for novel therapeutic strategies targeting splicing dysregulation, as we will discuss in the following topic. In summary, splicing dysregulation is connected to various diseases. Gene-based therapy, like exon skipping and genome editing, targets genetic defects. Pharmacological intervention aims to modulate splicing factors or improve splice site recognition. These strategies hold promise for treating splicing-related diseases. Alternative splicing is crucial in neuronal development, allowing for precise gene regulation needed for neuronal processes. It produces different mRNA versions with unique functions, influencing neuronal diversity and circuitry formation. Disruptions in splicing can lead to neurodevelopmental disorders, and understanding its mechanisms is important for therapeutic development. In summary, we have covered key aspects of mRNA splicing and spliceosomes, including alternative splicing, regulation mechanisms, disease implications, and therapeutic opportunities. This knowledge is essential to comprehend gene expression regulation and its implications for human well-being. Future investigations will likely unveil novel diagnostic, therapeutic, and preventive techniques for splicing disorders.